Okay. Assalamu alaikum. Good afternoon, distinguished guests and colleagues
Uh,
and
excuse me, Gulrukh. I call you. Kindly wait for a few minutes. Sir has suggested that more participants should join online, so we will wait for a few minutes.
Okay, ma'am. Okay, fine.
Thank you.
Yes, sir.
Guruk, you can start now.
Okay, sir. Assalamu alaikum. Good afternoon, distinguished guests, colleagues, and Mr. Javed Khan and Professor Excellency, Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General, OIC Comstech. So it is my pleasure to welcome you to the Comstech Secretariat. I'm Gulru, Program Officer at Comstech. It is my honor to welcome you to have arranged this workshop on metabolites and AI of diagnostic testing. So, to start the workshop with the permission of the chair, I request Mustafa Zaini, program manager at OIC Comstech, for some holy verses. So, please, thank you.
أعوذ بالله من الشيطان الرجيم أمن هو قانتنا آناء الليل ساجدا وقائما يحذر الآخرة ويرجو رحمة ربه قل هل يستوي الذين يعلمون والذين لا يعلمون إنما يتذكر أولو الألباب صدق الله العظيم.
Jazakallah. I would like to invite His Excellency Professor Dr. Muhammad Iqbal Chaudhry, Coordinator General of OIC Comstech, for the welcome remarks. Sir, please.
Thank you very much, Gulrukh. Professor Dr. Buddhis Israr, my colleague, and I see many of my colleagues there. Thank you very much that we have an important webinar today, which is delivered by Professor Dr. Javed Iqbal Khan. And he is actually affiliated with Forensic Assessment Consultant team. He's a forensic scientist, and he would be discussing metabolomic and AI of diagnostic testing. Now, this is an extremely important field. We understand that application of artificial intelligence, particularly data analytics and generative AI, is now widespread, particularly in different fields of sciences, and certainly in forensic science is an important aspect which he would cover today. I believe that we have many participants from different countries, and Dr. Javed Iqbal Khan is going to present an exciting lecture. Let me introduce our speaker today. Dr. Khan holds a dual Master of Science degree in biochemistry and molecular biology from the University of California, Riverside, and in biochemistry major with microbiology from the University of Agriculture, Faisalabad. With over 3 decades of experience, he has served as a forensic scientist at the California State Department of Justice Bureau of Forensic Services, Riverside, testifying in more than 500 cases across federal, state, and local jurisdictions of courts in US. He has held leadership role as the director of the laboratory operation as SDXRA, Alcala Diagnostic Clinical Laboratory, and currently serve as scientific forensic assessment consultant with the Tony Information Exchange Group and at the Utah University of Tony's office. In addition, his forensic expertise, Dr. Khan has contributed extensively to academic pursuit as an adjunct faculty member and developing and teaching core curricula in forensic toxicology, biology, and chemistry. He has also trained forensic team in advanced analytical techniques, including gas chromatography, GCMS, HPLC, LCMS, and FTIR, ELISA, and spectrometry, others. Most importantly, I think what we need to do in Pakistan, forensic sciences of different type, is not only that forensic is used for criminal justice or identification of bodies or whatever. It is involved in all kind of sciences, including toxicology, including food science, including identification of unknown compounds, and whole range of different disciplines in forensic is a key factor, particularly the expertise of Dr. Javed. Is important that he would be presenting here in the context of metabolomics, which is an exciting field where you are capable of identifying and mapping metabolites at a particular time in a very complex matrix. And by using AI tool, I'm sure that one can make sense of metabolomic profile in a very profound manner. With this, I would like to request Dr. Javed for his presentation.
Okay. Thank you, sir. Thank you. Okay, thank you. So, sir, please. Yes. Yes. Okay, sir, please join us for a lecture. Metabolomics is your, yeah, I think so. Yeah, thank you.
Assalamu alaikum, everybody.
Welcome, Salam.
Assalamu alaikum, everybody. I thank you all of you for giving me the opportunity to speak something about the new sciences here. I thank my old friend, he and I are both way back and during that program. And he found me from somehow last time I came to the United States a year ago. Then we got connected at this time and I told him I was coming. He told me if I can give a presentation. I had two choices of presentation. One was the forensic aspect and one was on metabolomics. My friend suggested that I should go with the metabolomics because it had not been for America in Pakistan at all. They're very, very new techniques and it's basically in America and Europe, we are still starting to surface of it. So we'll go through that. This is my brief introduction. My phone number and email address is right there. And I'm published author of my books. These two copies of these books are sitting here. If anybody wants to browse that. This book is used worldwide as a college textbook and university's textbook for the courses on forensic chemistry. This is these two are my recent books. This Admittance of Forensic Evidence as Credible Witness in Islamic Court. This book is intended for the people who understand very little about forensic, especially in our court law, and prompted by one of my few friends in judicial system here. And mostly this book focuses on 100% from Quran. I did not get any reference anywhere else but the Quran. And this book is the collection of my cases around my 22 years of experience as a forensic evidence, forensic scientist. In this book, I have about 37 very unique cases that were solved using very different techniques. So they are available out at Amazon. This is the pyramid of life. And if we look at that, we receive genetic information from our parents, mother, half father. then they transform into proteome and come into metabolome. Proteomic and metabolomic system, 99.99% depends upon three factors: physiological influences, environmental influences, and microbial influences. So basically, these three influences determine our metabolism, how our metabolism changes with the environment, changes with the onset of our what we breathe, what we eat, what we do in our life. And my whole talk will depend upon this one, how this changes around our body when we are not sick and when we are sick. Currently, our healthcare system is reactive. What I mean by reactive is we do not see the doctor when we are good, feeling good. Who wants to go to the doctor, right or not? We go to the doctor when we are sick. So doctors perform some tests, urine test, blood test, different tests, right? And then they diagnose. Some are diagnosed by diabetic, some are diagnosed for example, kidney, some are diagnosed by heart, depending upon what kind of ailment is, how we saw the doctor, which specialty we saw. And reactive diagnosis and reactive treatment. So doctor tells us that you have diabetes, you don't do this, you do this, you don't do that, you do that, right? Or if we have a heart problem, then they suggest something, medication, everything else to regulate our heart. The environmental impact for most of us are very different. We mostly overestimate the diseases, genetic contribution of diseases to our big side. Everything is genetics. Breast cancer genetics, colon cancer genetics, heart problem genetics, right? That's what we usually think. And underestimate the contribution of environmental impact on our body on a regular basis. Exogenous exposure of our body to external environment through intake, inhalation of food, pollutions, and chemicals create endogenous responses in our body. The whole metabolism changes with respect to our body response. Cancer is both metabolic disease and genetic disease. All the new or old drugs target metabolism, cancer metabolism. And this is the recent statistic. As you see, cancer type, hereditary impact, and contribution of environment. Lung cancer, 8% is hereditary and 92% is environmental. All these cancers you see here, more than 90%, other than leukemia, you see only 88%. Every other cancer onset of any cancer is environmentally impacted upon us. We don't think that way. We think because we got sick, or parents were sick, or the grandparents got sick, that's why we got sick. This is another type of cancer: colon, leukemia, bladder, kidney, testicular, head and neck. All of these are more than 90% impact on our body. These are the toxic exposome that people get exposed to. These are the deaths due to the toxic exposition. And if you see carefully, all these common diseases, common pollution that we have around us causes about 2.5 million deaths per year in Western country. I don't have any data for this America or Pakistan or any other country. So you can imagine the 2.5 million deaths due to all the environmental impacts. on the human being, human body. A growing number of approved diagnostic markers, cancers, or ongoing autism. And these are the markers that we target currently. I just wanted to make sure that these to highlight those which specific biomarker is used for the different types of cancer. And eventually this was our breaking point where we saw that 85 to 90% of death in developed world countries due to chemical exposure, and 15 to 10% is due to genetic. So it's a big span, big margin between the environmental exposure and genetics exposure that we have been blaming all along. These are the 11 molecules commonly used, consumed by us. And we, the scientists, went ahead and found in America, North America, and Europe, again, this data is European investment data, not the Eastern data or anywhere else. Good decay. Look at that. And I highlighted these with the, look at that, total 11. 7 of the 11 common diseases starts from sugar. Only sugar. You can imagine innocent sugar we eat all day. We think it's nice to have taste these things. Lipid, 1, 2, 3, 4, 5, 6, 7. 6. More than 50% of these diseases are caused by lipid, and 5% of these caused by smoke. So if we, not I'm saying completely eliminate 100%, what I'm saying is if you regulate the intake of these two, mostly these two compounds in our body, sugar and lipid, we can eliminate these common diseases in our body, basically. That's what it boils down to. And if we take care of all these, we don't smoke, we take sugar wisely, and we take lipid wisely, we can completely eliminate the 11 common diseases in our body just by doing that. I'm not saying completely stay away from sugar or fat. All I'm saying is if our intake is regulated or watched or observed, it helps. The outcome is that whenever we have genomes, they are working in our body, or they will work in our body, tell us what could happen to our body. Like we inherited, for example, breast cancer gene. It happens, we are told, okay, you could get this kind of disease, for example. You could get breast cancer if you're a female, right? Or if your parents or forefathers have heart problems or any lungs problems, then we can say you could get that. Transcriptome is what tells us what appears to be happening in our body at that given moment. Proteomes tell us what makes it happen. And finally, metabolomics tells us what will happen negatively and how these negatively affect our body on a regular basis. So basically, metabolomics tells us what we are doing to our body will eventually negatively affect us. It has not impacted it yet, but we are expecting that it will, or we are predicting using AI that it will happen. Future of healthcare will be proactive care. In that, before we get sick, then go to the doctor every year or every given span of time, they will go and do the urine, blood, serums, all these tests any of these, or all of these, or some of these, they will test it. And proactive testing will tell us what not to do to our body any longer. How can we prevent it from doing happening? So this is a preventive actions and proactive action for our body. Metabolomics are more associated, and this nomenclature, and if you look at that, mostly the technique that is used goes here and or L. So in this case, don't worry about, but mostly commonly used is metabolomic. It is a quantitative measure of the dynamic multimatrix metabolic response of the living system to pathophysiological stimuli or genetic modification. Definition is very simple, that each body organ respond differently to chemicals we are exposed to. And that's there. And these are only the small molecules that are responsible, not the bigger molecules. So far in healthcare, we have been ignoring these small molecules. We think they are insignificant. They're small children, they're small, they don't know nothing. These kind of exposures that we have, what would happen if we continue doing what we are doing to our body? And this technique would become an AI of the human health. So, in other words, this case is own technique if it is implemented, and it's a very early stage for now. 99.99% individuals in America and Pakistan have not even heard about the word metabolomics. So this is last year I came and I gave a lecture on that, not Japan, and this time I'm giving a lecture on a different other areas, including care, to make you aware of the upcoming techniques. It represents the, instead of now we go, for example, we do one test, two tests, depending upon what kind of ailment we have. If we go to the doctor as a diabetic patient, what does he do? Test blood sugar or maximum A1C. In this case, we are not testing only one metabolite, we are testing fragments or small metabolite in our body. And the response of this will reveal a set of biomolecules that are being secreted and differentiate from the normal and the abnormal individuals. And once they are abnormal, the individual will be able to tell that this person is sick or this person will be getting sick. And we will I have some examples for you. I will show you along the way. So that represents one aspect of cellular function. And as we know, cellular functions are different. Genes, VNTR, STR, these produce the metabolite fragments. They produce dinucleotide, trinucleotide, mononucleotide, and these are called metabolomes of DNA. Transcriptome, microRNAs, metabolic fragments, or nucleoside, nucleotide, monoditride. And these are metabolomes of RNA. Proteomes, hormones, every type of proteins, enzymes, metabolite into amino acids, natural, unnatural, ditripeptides, or metabolomes of protein. Lipidomes, metabolite fragments of fatty acids, lipids, natural, unnatural, or metabolomes of the lipids. Transcriptome transforms into genomes, goes into transcriptome, goes into proteomes or lipisomes. Since the metabolism of each tissue and organ are specific, metabolomes of each organ tissues are unique and are identifiable. Because our kidney metabolize lipid, less lipid and more water soluble, correct? Our brain metabolize sugar more than any other metabolite. So likewise, these will generate different metabolomes. Each organ will have a different set of metabolomes. Kidney, liver, heart, pancreas, lungs, every brain, every will have a different set of metabolomes. Let's talk about the history of that. Introduced by Dr. Roger Williams in late 40s, so it's not it has started with the long time ago, but it has not been this level that we are now today. They developed metabolic profile or metabolic pattern. They were not known as metabolomes at the time. Started from 40s, going to 17, then we go into 74. Steve demonstrated the role of magnesium in ATP complex. And 84, Nicholson showed NMR spectrometry by the diabetic mellitus recognitions. 1994, LCMS metabolomic experiment were performed. In 1996, another metabolomic performed that they recognized that oligonucleotide was observed as sleep inducer. In 2005, first metabolomic tandem mass spectrometer-based characterization human metabolism was developed in the laboratory at Scripps Research Institute. Up to this one, 27 until 2022, all this was based in as a part of R&D. It was not publicly known, it was not publicly introduced because it was just R&D, research and development, research and development, research and development. Dr. Richard, he contacted me because he wanted to transform this R&D into diagnostic laboratory. So he contacted me in 2022 to help him get his R&D into technical testing. So I got involved in that and helped him get ISO 17025 and ISO 17448 for 15148. After retiring from the Department of Justice, I started consulting process as in ISO International Certification, HAP, College of American Pathologists, and Forensic. So this consulting, that became my introduction to this. I didn't know anything about that before. So we helped University of Edmonton and Jewish Hospital in Montreal, Canada. I was there last month in Montreal to help this laboratory get accredited. So when they got accreditation, other laboratories who were doing also the same metabolomic, they asked Dr. Richard how he got the accreditation, so he named me, and this laboratory contacted me in 2023. CLIA certification for Texas Hospital of Testing and Kidney Function Deterioration for Diabetic Patients using metabolomics. This was a very interesting experience. Dr. Sharma, an Indian descendant, he developed a method in that the kidney of the diabetic patient deteriorates. So this test basically tells the patient your kidney is deteriorating and help yourself. So they have this test, this qualitative test. It gives you the idea that your kidney is stable, unstable, negative, or positive. Nothing wrong. So this test is being implemented after it is approved from we got the license now. All we are doing is waiting for the validation of the method. Validation of the method, this type of method require about a year for validation. So Sharma is doing that. After that, you will be able to done. This is another laboratory, Laris. They contacted me in Massachusetts, and I helped them. This is also kidney post-transplant function. For example, if person gets a kidney transplant, and these are the websites you can check on these ones as kidney transplant for this one, Laris Laboratory. It tells the doctor whether this patient is patient body is accepting this kidney or rejecting it or deteriorating. This is another laboratory, our own laboratory in Connecticut, is testing new eye function to understand the development of different polyps in our body, in our gut. So, and this lab, Jewish Hospital in Montreal, is also testing high metabolite for cancer detection. So this is before the cancer is on site, before a person is suffering with cancer, this laboratory. So these are the four laboratories at this time in the whole world. They have their ISO certification, they're taking the next step into getting into the new era for starting the diagnostic testing in metabolomics. But problem with this metabolomics is since it is environmentally induced. The data so far in Pakistan or in our third world countries, we don't have any of our own data, because we don't have money to do R&Ds in data. So what we do is we take American data and we implement here for our sugar testing or for our any other testing in our laboratory, any diagnostic testing we do, or any reactive testing we do for heart panel, for kidney panel, for liver panel. All these testings is done. We are using international data, and international data in that case is usable. But here, since it is induced by our body with the exposure to our environment, with the exposure to our food, we cannot use Western data. So they are developing the data. So far, this is a year old, so it's up to years old. Last year it was 9. It's a little bit less than 1 million. I'm sure it's more than 1 million data of MS of different metabolites developed by Sweetpick Lab, LCMS data, this human metabolome, Dr. David Wishart. So he's also developing the database, and this is also a year ago. So probably they have developed the data. So in that case, once they develop the data, then they will share, and they're developing the data for the normal people. So because we cannot tell the person is sick or will get sick unless we have normal data. Because normal data we use for comparison, right? How do we know this person is diabetic or not diabetic? Because we have the data. right? We compare it with that data. We have developed normal ranges, and below or above normal ranges is the event. Likewise, any other test that we perform right now, we have the data, we compare it with the data, and we can tell this person has a normal results or abnormal results. It's another part of the HIM data base that is being developed. Purpose of this database is to create a normal function readout in our around us for our physiological state. It will become a kind of our fingerprint, our DNA, specific for individual to individual, because our metabolism, our food intake, our environment is different. That can be used to predict the difference between normal and abnormal tissue, organ system, function, etc. The type of metabolomics could be fat soluble and organ associated with the fat soluble, or these system associated with fat soluble, or these specimen tested or blood, serum, spinal fluid pieces. Water soluble, organ, tissues, kidney, uterus, urine, genital system, specimen to be tested, blood, urine, spinal fluid, and feces. These are the techniques that we use: LCMS, GC, GCMS, GC, LCLC, LCMSMS, NMR, or any other genetic analyzer can be used for the test. Specimens are used as the way we are using now. There's no difference. These are the equipment we use. Now, here is the very interesting slide that you all see. Our body secretes hundreds of thousands, if not millions, of small metabolites in urine and feces, right or not? In our blood, so many. How to find the right molecule for the right reason, right or not? We don't know that at this point. That's why it's separated. Whenever we do the testing, it is separated for targeted versus non-targeted. And targeted or focused later on, and non-targeted. Just give me a simple example. Pacific Ocean. If you want to go to Pacific Ocean and find the fish of your interest or choice, how many chances in how many million years you will be able to find it when you put your card in? How many things? It's very astronomical right now. So likewise, our response is astronomical. That's why it's very, we may not be able to see the outcome of that. Our great grandchildren probably will benefit of. But that will increase at least 50% of our age limit from now on. If a person is 80, add 40 more into that, and then it will be having a normal life. Now, unlike the regular testing, this is the data analysis of that, and this is the data that we generate. You see how many metabolites there are, and then this is the spectra of that. And this will be the outcomes, statistical analysis of that for your AI effect. That will particularly tell us where is the problem and how it is doing. And I'll show you a few results here. This is, for example, adult kidney transplant rejection. Matrix is urine, and 9 metabolites are tested in that, and the spectra will look like this way. Pediatric kidney transplant rejection, pediatric urine samples is used, and 4 metabolites are tested on this. This is the difference between systolic heart failure versus diastolic heart failure, adult serum is used as sample, 4 metabolites were tested. Chronic fatigue syndrome, 6 metabolites are tested from the serum, and the data will look like that. OEO, pediatric urine sample, and 3 metabolites are tested, and data will look like that. Colon polyp that I told you that Aero Laboratory is doing. So they are doing 12 metabolites from adult urine sample, and colon polyps will be identified that this person is developing colon polyps and have to be removed. Once again, reactive testing techniques are physiology dependent, and worldwide diagnostic data and testing techniques can be shared and are accepted. On proactive testing AI healthcare, response is dietary, bacterial, viral, and environmental dependent. Response of human physiology varies from area to area. People living in Islamabad will not have the same data that people living in Karachi or in Peshawar. They will have to develop their own data. Testing technique, however, can be accepted, but the data will have to develop our own. And that will create a problem for implementing in our countries because we do not have any data. And developing a data, getting 5, 10, 20, 30, 40,000 normal patients and doing R&D and applying the data versus sick, between sick and the normal people will have a big impact on that. So I don't foresee any implementation of metabolomics in Pakistan or in other third world countries or Asian countries for next maybe 150 years or so. Any question? Any question?
Dr. Sir, I really appreciate your talk. That is really enlightening. But I want you to comment on the last statement which you said, that you're not seeing any advancement coming for Pakistan in the next 150 years.
Yeah.
Is it only because of the funding or because of the lack of expertise?
All of the above.
Or
Yeah. And first of all, we don't have funding. Second of all, we don't have the kind of know-how of how to collect data, how to put the data together, how to create it. We don't have any data set for anything. We have individualized ourselves. And unless we have data for normal people, we don't know how to test the abnormal people.
you're very right with the very high pollutants in the air.
Yes,
Our data must be very different from the rest.
exactly. Because, and still, we don't know whether we have to have this data 5-mile radius, 50-mile radius, 100-mile radius. It may vary. And this is also still infancy in America as well. We do not have that many labs who are doing metabolomics at this time.
And besides genetic makeup, also there is role in that.
That's correct.
Dr. Javed, I have a question. It's very eye-opening discussion details and how we can go to right? But there's one debate we won't follow her, that actually our cell's life is 150 years. And it reduces when due to the
And
environment.
visual, that, yeah.
How you are going to deal with it, whether it is a metabolic thing or whether to the environment or whether due to the eating habits of these things. So how you take it according to the metabolics?
It depends upon lots of factors.
The characteristics. can you predict by the metabolic data that how much lifespan a person is going to have?
The lifespan reduces with the ailments, with the issue, right or not? If you're, some people are more sick, they die early. Some people are, I'm not talking about somebody going in the car and have an accident, that's not that.
Biological death.
Normal, right? Right or not? So in that case, we don't have any clue. But one thing is there, if that 11 common diseases that I discussed, if we regulate those three molecules, scientists are predicting that we'll add 50% up or ahead. I mean, the lifespan.
So why don't we go to the easier way to control it, three things and leave the other things and take it into?
I wish we could. I wish we had that much money and that much resources. Remember, science doesn't go jump from nothing, 0 to 100% one day. If you look at ourselves, we are old enough, I'm not talking about these kids, we are old enough to understand that internet didn't exist 30, 40 years ago, right or not? Cell phone we did not have cell phone. But the technology evolved, came, and we got where we are standing today.
I also want to hear some comments of you about whether you didn't talk about trace metals and nutrients, which is very important when you talk about the environment. How they affect these metabolic things?
See, it changes because we have regular normal flora in our body, right or not?
Yes.
Right? And normal flora react to what we eat. Let me give you a simple example. A home, happy home, the children are happy, right? Because they see and they feel the happiness, parents are getting along. In a stressful home, children are stressed. Why children are stressed? Because their parents are implementing stress upon them or imposing stress upon them.
See, why am
Likewise, our normal flora is our baby in our body, right or not? When we put these normal flora in a stressful environment, they get stressed and they respond differently than they would do in normality.
See, why I'm asking this, because you talked about metabolic things, and if you talk about the human body and you see the metabolic things, they are accelerated by these four enzymes, and which are very important, goes with the drugs like saying copper and all these things. So do you study these trace elements besides the metabolic proteins or?
No, only these are heavy.
Okay, so
How can you differentiate that this disease is because of metabolism or because of genetic?
Well, you can do that. You can, yeah. We are advanced
Just
enough to detect that, yeah, we are advanced enough.
Properties are the fifth of country of the world. Majority of people live in the villages. For these tests, they cannot afford them. That's all the difference. Even they are sick, they have no money to credit the cheap. Not in nowadays. It is very difficult to in Pakistan.
No, in any third world, countries like that, not in the world. Any other question?
Thank you so much for such a nice presentation regarding to metabolomics. I just want to know, as you mentioned at the last of your talk, that we might be unable to, after a long time, they will be unable to run this facility in the integrated specific process. Would you like to give some idea regarding to how many countries they have such type system regarding to metabolomics, either that they can?
Europeans and America and Canada,
They have.
at this time. They have not started the metabolomic laboratories yet, but they are doing R&D and data. Because this is the process of data for now. But very few, I gave you 4 or 5 laboratories, they have evolved from R&D into ISO certification, and into ISO certification, into CLIA certification. CLIA certification is in America, ISO certification is internationally. So what they are doing is they're trying to be ahead of the curve And they have lots of investors who are ready to invest upon their research and upon their testing.
So there's the last question.
No, you don't have to have last question, you can have second one.
Okay, second last question. So how we compare it, if we take it as a diagnostic tool in the future, how we compare it, work done by testing through iris and things like that, because it still is that it needs a wet lab and testing all these things, and even you have mentioned that NMR is used. So for me, it will be very good if they are going in the future by iris.
Yeah.
So what do you think about it? So it will be easy for practice, this biodynamics to NMR and other things?
No, it depends upon, see, again, it depends upon lots of, NMR is used for a specific type of a metabolite. LCMS is used for specific metabolite. DCMS is used for specific metabolite.
But it won't be easy to do it with the.
No, it's not, there is no easy or hard way. There is way, because look, molecules you're looking for testing dependent. Yeah.
Okay.
Sir, I wanted to ask that, as you mentioned, there's a very large sample size. Even if we compress it down to a single organ, there's a very large sample size and we have to differentiate it into quantitative and non-quantitative.
Yeah.
So how will we perform all these tests in a small laboratory? Can we compress it down and can we make it affordable for patients?
We will eventually, everything will be affordable. Everything starts from very expensive. If you are not old enough to understand, if you guys seen old James Bond movie, they had big cell phone in their car. This makes sense of James Bond, we say, wow, right? Now everybody has a cell phone in their pocket. Because it was, at that time, the cell phone was $40,000 in per car. Now, cell phone is in your pocket.
So in the future, it will be possible.
Everything gets, because we find ways to ease our techniques to make it less expensive, more affordable. Because it's not the money, for example, if you have $40,000 phone, will you have a phone in your pocket? No, right? So affordability is the more they sell the phone, more money they will make, right? So it's affordability. A $50,000 phone, James Bond phone, only the movie stars at the time used to have it. And common people could not afford it.
It is something similar to the DNA sequencing.
Yeah.
Like it was very expensive back then, and now it is.
That's right. DNA sequencing was very expensive. Now DNA sequencing is easily available. Same thing with this one as well. Any other question? My books are here. If anybody wants to look at that. This is not that I'm giving it here. I'm just leaving it here for you to see that. And they are available at them.
So we have some online participant question answers. So
Yes, please.
okay. Sir, the first question is by Irfan Ahmed, and he's asking, sir, the causation of a condition by environmental factors depend on how the human body react to those factors. If a condition eventually occurs, such as breast cancer, it may lead to alteration at the genomic level. In that case, correction at the genomic level would be required. Do you think such genomic correction could actually help reverse or prevent the disease?
That's our target. And that's our target is to make sure to have a preventive medicine rather than, and preventive medicine will not be the tablets or injections or those things. Preventive medicine is don't eat this and don't eat that. That will be preventive.
Okay, the next question is by Amara Sabir, and she is asking, how do researchers identify unknown metabolites in a sample?
That's easy because we have a database of different molecules, different metabolites, and all we do is data search. That's why the database, development of database is very important.
Okay, so I want to check the next question. Okay. The next question that is by the Sami Khan, and he's asking, if we project 5 to 10 years ahead, how do you see AI-driven metabolomics transforming preventive medicines and early disease detection on a global scale?
Global scale, you cannot compare our country with the global scale. Our
Yeah.
country is way behind. Remember that.
Yeah.
So in that case, just multiply all these years by 10 or 20 or 50, then we'll see about implementing of this technology in here. That's why I said maybe your grand, not my great grandkids. My grandkids are already here. I'm talking about your great grandkids will have the benefit of that.
Okay. The next question is by Mohsin Ali, and he is asking which mass instrument you prefer for metabolomics.
It's not a one generic method. You
Okay.
can use any equipment technique depending upon what you're looking for.
Okay, sir. Okay. Okay, the next question is by Shakirullah, and he is asking what types of tissues sample can use metabolomics?
As I
Okay.
gave you the blood, urine, feces.
Okay.
Yeah,
The question is by the Rakib, and he is asking what are the limitations of current AI models in analyzing metabolomics data for clinical use?
there's no limit. Limit is basically on our mindset at this time.
Okay, so agree? Do you snap? Okay, thank you. Okay, the last question that is by Sami Khan, and he's asking, do you envision metabolomic AI diagnostic being used in real-time clinical setting, such as point of care testing or continuous metabolic monitoring?
At point of care testing.
Okay, fine. So yeah, thank you. Thank you. One thing I want to add in as Sir Career Life Sir is also establishing the state-of-art National Forensic Lab in Islamabad. So yeah, that is the one thing I want to add in, Sir.
Yeah, I have been invited by Interior Ministry to set up the National Forensic Laboratory. This trip is paid for, and I'm cheating on them standing here. Because I told them I'd be available for other laboratories. That's what I'm here for.
Okay, sir.
That's wonderful.
Okay, sir. Thank you, sir. Thank you so much, sir. It's a very valuable lessons we learn. So I think we must have a claps. Yeah. That's all. That's it. Thank you. I would like Sir Ismail Diallo for the closing remarks. Sir, please.
Assalamu alaikum. Thanks again, Doctor, for the high-level presentation. And the presentation provided better understanding of the role and importance of metabolomic in the diagnosis and treatment of disease such as cancer. The second point is it provide also insight into the role of the environment and yet in the onset of certain disease. The combination of metabolomic and artificial intelligence opens a promising path for precision medicine with greater precision in diagnosis and treatment in personalized medicine. Despite the reluctance, this revolution is underway for a healthier global population and a reduction of the cost of patients' care and treatment in the world. Thank you again, Professor, and congratulations for your for accept to come here. Dr. Sam, if you have something to add. Okay. Thanks, everybody. Definitely.
So yeah, sir, with you. Okay. Yeah, they are already.
You take that.
Okay, yeah. Before we go ahead for the closing ceremony for certificate distribution, I want to add some details of Sir Ismail Diallo. He's the deputy from Senegal and he is now working here as the OIC Africa Regional Advisor at OIC Comstech. And he worked in a ISESCO, the Islamic World Education Scientific and Cultural Organization, in charge of strategic planning, development, and implementing in research projects in field of different agriculture, forestry, and environment. So thank you, sir. So I would add to sir, thank you again for your answer. Thank you. So I invite Sir Javed and Mr. Diallo for the certificate distributions. Okay. Okay, I also okay. I also invite Madam Mudassira Sati. Yes, please. Okay. And sir, also sir, yes, please, for giving certificates to participants. Yeah, thank you. First, yes, we are. Someone but we.
डिस्ट्रीब्यूशन भी बता के जाना साथ साथ अगर इनके पास है तो
Fatma.